Hello, I'm Ibrahim Imam, and welcome to this episode of the Neurology Lounge podcast, where we will continue our exploration of motor neuron disease. In this episode, I will be speaking to Professor Kevin Talbot to discuss the management of the disease as conveyed in his book, aptly titled Motor Neuron Disease. Kevin Talbot is Professor of Motor Neuron Biology and head of the Nuffield Department of Clinical Neurosciences at the University of Oxford. He's a clinician scientist with 25 years of experience of diagnosing and managing MND and related diseases. So it kind of spans the scientific and the clinical aspects of MND. He leads the neurodegeneration and cerebrovascular theme of the Oxford Biomedical Research Center. And the main focus of his lab's research is to improve clinical models, preclinical models of ALS, which is amyotrophic lateral sclerosis. And he focuses on the early stage of the disease. His lab has identified disease-specific phenotypes in motor neurons, which provide the tools for screening drugs of potential therapeutic benefit in ALS. His research is also closely linked to work being carried out by his Oxford colleagues on biomarkers with application to experimental medicine studies, and that's aimed at accelerating translation of promising drugs. Professor Talbot, welcome to the Neurology Lounge podcast, and I'm most grateful that you accepted my invitation.
Thank you. I'm looking forward to talking.
Clearly, from your bio, it's obvious that you're kind of in the translational field of MND. Is that a motivation for writing the book, or did you have some other reasons behind it?
I think the key motivation for writing the book was that a motor neuron disease clinic, particularly in an academic setting, there is a huge amount of information transfer. When somebody's diagnosed with motor neuron disease, obviously everyone's different, but most patients, feel they need to understand what's going on because something terrible is happening. They've been told they have a disease that's progressive, is not curable. And so that requires some understanding. And people have become increasingly sophisticated in their need for knowledge. And of course, they can get information from the internet. But I think that the information we have to offer them in the clinic is not really easily available on the internet, and a lot of bad information on the internet, and people are vulnerable. So one of the key stages in people coming to terms with the diagnosis is having a better understanding. So I think I wanted to put into a book what was in my head and what I say to patients every day, so that people could read it in their own time and reflect on it and select information.
And it's a co-authored book. How did you manage to get your ideas all kind of in sync?
Okay, so I have to admit that this was written quite a long time ago now. Yeah. So I don't think I can remember the details of how we wrote it. Okay. But I think, essentially, this sort of... So I wrote it with my colleague, Rachel Marsden, who is a specialist nurse consultant in motor neuron disease. So we've been running the clinic for 22 years. And she has a profound knowledge of the patient journey and how you care for people and symptom control, as well as, just understanding the disease in a very deep way. So she I think, is able to write sections on management and caring and supporting. I think I wrote the stuff on symptom management with medications, as well as the basic what is motor neuron disease and how does it arise kind of stuff. And then we swapped chapters and, revised it together. So I think, chunks were written independently, and I took overall control for trying to create a single style.
Okay, because there wasn't any evidence of a conflict between all the chapters. Let's start with the basics. MND and ALS. There's a bit of confusion, I think, across the Pacific as to...
Well, there is, and it's not very helpful.
No.
Personally, I would abandon the term motor neuron disease completely.
Okay.
Because, there are a whole variety of reasons. One reason is the rest of the world calls it ALS, so it's just unhelpful to be calling it something else. That confuses people. Okay. ALS describes anatomically what is going on. There is muscle wasting, and there is lateral sclerosis, meaning scarring of the corticospinal tract. So this is a bit of a sort of technical justification for abandoning the term. It's not a disease of a cell, it's a disease of a system.
Okay.
There are more than motor neurons involved. There are glial cells and other support cells. There are cortical cells that aren't motor neurons that are involved. So, and there are many different motor neuron diseases. So there's spinal muscular atrophy and Kennedy's and lots of things. So I think calling it motor neuron disease is not helpful. It's not accurate. If the rest of the world, if everyone decided to call it that, that would be fine, because we just simply should have the same term. Otherwise, I think I used to find that, and this is less true now, people used to come into clinic and they used to think if I said they had ALS, that was the worst kind to have. Well, ALS and MND are essentially synonymous. So I don't think we should really be preoccupied with, distinguishing the two. ALS and MND are used interchangeably.
Okay, so that kind of limits the variants or the subtypes to the PLS and.
Well, so the classical subtypes, I think that's breaking down that kind of that nomenclature. A student textbook will have there are 4 types of motor neuron disease. Well, I don't think that's true.
Okay.
That's not how I think about it. I think there's amyotrophic lateral sclerosis, and then maybe PLS is different, maybe it's not. Otherwise, I don't accept that PMA, for example, is a thing, as a separate entity. It's simply lower motor neuron predominant ALS. So if you look at people with PMA at post-mortem, they have pathology in the long tracts, always. So, this is a condition where you have damage to a distributed system, which has an upper motor neuron component and a lower motor neuron component, and that damage, for reasons we don't understand in different patients, is reflected more in one end of the system or the other, or it's equal. But that's why it looks different, but it's not a different thing. And I don't think that anyone can come up with a consistent definition of PMA, for example. That means that everyone around the world is talking about the thing in the same way. So I don't actually think the term's at all helpful.
Okay. Well, that's going to be revolutionary for my website. I'm going to have to change the terminology.
Well, at some point, I'm going to write an article explaining that. I mean, not a lot of people don't agree with me, but it's a bit philosophical, really. I mean, if you and I think that there are patients coming to our clinic and we want to call them motor neuron disease or ALS or whatever, our task, if you want to understand that condition, is to decide whether they will have the same thing or something different, right? As a scientist. So you start, wanting to break it down into subtypes because you think that might give you a better understanding of what's going on. But if you do that on purely clinical grounds, I don't think you necessarily get to the bottom of the biology that is driving that variation. And in fact, I tend to think of an individual patient in front of me as having a form of ALS that has 8 to 10 different dimensions. And the kind of dimensions that are important are age of onset, rate of progression, site of onset, whether they have or do not have respiratory involvement, the relative upper and lower motor neuron burden, the presence or absence of cognitive impairment, the rate of progression from one anatomical zone to another. So, for example, does it move from your left leg to your right leg or from your left leg to your left arm? Those are different types of disease, I think. So, you end up with about 10 different ways in which you describe somebody. And on the basis of that, I think you can have a very good understanding of their disease trajectory, their natural history, their survival, and things that patients want to know. So that's how I think about it.
And I mean, from the surface, it looks like that is more likely to lead us to precision medicine rather than the old way of looking at it as MND.
Yeah.
Okay. Then the other confusion is, I suppose that's kind of American and British spelling of neuron and neuron with an E.
Well, I need to be careful here. I could bore you for hours on this topic. I mean, the word neuron or neurone is how it should be pronounced, comes from Homeric Greek.
Okay.
And if you take the Greek letters, nu, epsilon, whatever, I mean, so it's neurone. And that's in ancient Greek, it was pronounced neurone. Okay. And it means sinew. So when they were hacking each other to death with swords, the cords that they saw, which are probably tendons, or maybe they were nerves, they called them neurone. So then Waldeyer, a German anatomist in the 19th century, who was not a neuroscientist, coined the term neurone for nerve cells because he thought they resembled these cords that he read about in Homer. Okay. So that's where the word comes from. Now the French, because they wanted to be precise, added an E because in order to make it sound neurone in French, you need an E. So English sort of picked that up or didn't. In the 1930 edition of the Oxford English Dictionary, it's neurone without the E. That's the correct term. But everyone adds an E occasionally. Then the Americans took it off and say neuron, which is completely wrong anyway. So there you go. I could carry on, but that's very boring.
But you prefer, obviously, then without the E is kind of
I
more.
do, but it's not something I'm going to lie down in the middle of the road to defend. I mean, I think it's just a preference, but I prefer it without the E.
Well, I've had quite a few people picking me up on this on the website. I use the E, and the kind of people are very strict about. They don't want to see the E there. So
But, strictly speaking, the E isn't necessary if you're speaking English. But the point is, and this is the point everyone misses, you pronounce it neurone, not neuron.
That's very helpful for me personally. You wrote that the reason particular people get MND is a mystery, and yet there's so many risk factors. Is From what you've said so far, is it likely that there's a final common pathway for all these different risk factors leading to MND?
Well, so I can answer that in two ways. I could say probably not, in that there's so much going on biologically. It depends what you mean by pathway. So the essence of motor neuron disease is that, the final common pathway is the anatomical and physiological connections. It's that's the point of convergence. So you say, why does the condition exist? It exists because that network has a particular vulnerability. Now, there are many different upstream biological triggers to that, I think. Obviously, aging is a key trigger, because even if you carry a mutation from conception, you have to get older. So simply having a mutation is not a sufficient condition to have the disease. You have to get older. So the combination of genetics, whether it be a single strong genetic effect or a combination of genetic effects, the way that we develop, the way the system architecture is laid down, the way it's used in life, the way it ages, this is what ultimately leads to some initiating event, which we don't understand at all. So I don't think that is ever going to be down to one thing that you can say, well, people get motor neuron disease because this particular pathway was triggered. I think when you try and model the disease, you see a vast array of different things happening. But we don't really have a way of understanding what the initiating factors are, which is really a key element to definitively trying to find treatments.
And this link with aging, does that mean we probably would not be able to resolve the disease without sorting out what leads to aging?
Possibly. I think what the optimistic way of looking at that is saying, well, if aging is a cofactor and the median age of onset 64, if you understood what biological processes in aging were intersecting with all of the other elements, and you modified those, you could push the age of onset to 84 or 94 or 104. Now, you might think that doesn't solve anything, because ultimately, if someone has a disease that lasts for 2 or 3 years, well, they have it when they're 80 or 60, it's the same level of burden, distress, etc. One way of looking at it is simply saying that you'll be giving people decades of healthy life that they don't have. So that might be a good thing. You can look at it whichever way you like. But I think I think my view is that treating aging, that's a whole nother major problem. I don't think it's possible, in any kind of foreseeable time frame to do that. So the only way really aging would be modifiable is to understand that there are some very particular aspects of aging that affect the motor system compared to other systems in the brain. And I think that's currently very unclear.
One of the uncertainties. I always thought MND or ALS affected men and women equally, but the ratio you gave was 1.5 times more frequent in men.
Yes, it may be a little bit lower of a ratio, but it certainly affects men more, probably 1.3 or something like that to 1 ratio. And that's very consistent across populations wherever you look.
And as women live longer than men, I suspect that difference is even more stark, I presume.
That's true, yeah. So if you're a man, there is a slightly higher risk of getting ALS. That's an observation. So the question is, what is the biological explanation? And the type of ALS is slightly different in men, probably. So that excess is mostly accounted for by younger men with limb-dominant, lower motor neuron dominant disease.
Okay.
I mean, there are all sorts of different types. That is the sort of general trend.
Okay.
And so if you look in the postmenopausal period, then the ratio kind of goes to 1-to-1 pretty quickly. So this is something which is accounted for by a younger group of men, and maybe they have something different going on. I don't know.
And that's kind of slightly different from, say, Alzheimer's disease, which is predominantly female, isn't it?
I guess so. I'm not sure I know the figures precisely, but I mean, that would definitely relate to what you said about the demographics. Clearly, women are living longer. They're much more. I mean, if most people with Alzheimer's numerically get it in their 80s, and most men, died by the age of 82, and women are still living out onwards, then that probably accounts for that. But I don't know.
And then the site of onset, most people getting it in the limbs. Is the risk of developing MND or ALS in a specific part of the body related to how much we use that part?
I don't think so. I think that if that were really true, you would see, a lot more right-sided onset disease. So in the lower limbs, there's no relationship. We looked in the upper limbs at handedness and site of onset. So we asked patients who had, so a third of patients will have onset in one upper limb. So we asked them whether they were right or left-handed and which limb, the onset was. And we got a very small excess of dominant limb onset. So that's plausible because I imagine that the wiring diagram between the cortex and the cervical spinal motor neurons that lead to hand function is different in each hemisphere. That there must be some basis for dexterity being, and some people are very dominant. So, if it's something to do with the way we're wired up, and the wiring diagram for the right hand is different from the left hand, then it's plausible you might get more vulnerability in the right side. So there's a small signal when you look, but it's pretty small.
And how does that feed into this split hand syndrome where it's more likely to start on one side of the hand than the other? Is it kind of
Split
a
hand.
similar mechanism?
Yeah. I am less of a split hand enthusiast than many of my colleagues.
Okay.
It does seem to be a thing, but I think it's pretty inconsistent. I mean, it's been reasonably well studied, but by people who are very wedded to this idea of cortical driving disease. And also, their claim is that the split hand tells you it must be something to do with cortical organization. Well, the organization of hand function at spinal level is very complicated, actually. And it is possible that, that laterality is maintained in spinal cortical circuits. So I don't know. I mean, I think, there's enough evidence that it might tell you something about this being a top-down condition, but only in those people who have, involvement of their hand early, and only in those people in whom they have a split hand. But there are plenty of people who have upper limb onset who don't have a split hand, in my view. So it's not a consistent feature.
It adds to the complexity.
Yeah.
And then the spread, it kind of starts in one place and moves to another site. Is
Yeah.
this is a bit controversial? Is this supportive of this prion-like hypothesis of neurodegenerative diseases?
Well, so first of all, I think, we use terms like site of onset. And I mean, all we're simply saying is clinically, if you say to a patient, what was the very first thing you noticed, they have an answer.
Okay.
So does that mean that, something started in a location and then is moving from that location? Well, I think there is some evidence that's true, but it's not absolutely, definitive. But let's assume that that is what's going on. Then the implication of that is that, because actually, the spread clinically is not random. Somebody comes with their right hand involved, that the next thing that's going to be involved is their left hand or their right leg. It's not going to be their left leg. So if that is spread, it's a different type of spread in each case, because it could be, if you're moving from one arm to another, well, you can kind of imagine it's lower motor neuron predominant. You can kind of imagine that there may be something moving between the areas of the spinal cord. I mean, there are connections, reciprocal connections, or is it that whatever happened to the right side is now happening to the left side because, it's just falling over like a sort of set of dominoes. The vulnerability is there and in the other hemisphere, not without any connection. If it's moving from the right arm to the right leg, well, what sort of spread? What do we mean by spread? If you visualize it, then, well, those lower motor neurons are quite distant from each other. If it's upper motor neuron involvement of a lower limb, maybe there's a cord. I mean, so actually, the more you think about it, the more you think, well, actually, I don't think that this is easily explained by transcellular spread at all. So I'm skeptical about this in lots of ways. And classical prion disease does not obey anatomical and physiological boundaries. So if somebody with CJD comes to your clinic, what you see is the brain melting away because it's basically the protein is spilling out of cells and spreading not across discrete functional and connected networks. So it's transcellular. So what we might be dealing with here is transsynaptic spread. That is one explanation, possibly. But, it doesn't, clinically, this doesn't look to me like prion-like spread. To other people who don't think about these things enough, it might do. And these are often people in the laboratory looking at mice. And if you say to them, well, actually, I don't think that's what I think is going on in clinic. They are so keen on the idea that they don't want to listen to that. We've got to be very critical. I think it's been, it's lazy science just to sort of say, oh, this is a good idea. It must be true. What's the evidence for it? Well, clinically, I think the evidence is lacking. I think that the preclinical evidence has not yet been convincingly produced. In order for it to happen, you need a certain kind of biochemistry. You need a protein which is susceptible to templating. So we've been trying with TDP-43 to make this happen. I can tell you it's not easy. And this is a protein whose physiological function is to aggregate as part of its role in the cell. So, there's a lot of complexities here. I'm much more open-minded than I might be sounding here. But the reason I take this approach is because over 25 years, what I've seen is these kind of light bulb moments, going off. It's excitotoxicity, or it's mitochondrial, or it's this, that, and the other, or it's apoptosis, or it's, autophagy, whatever. I mean, there's no evidence for any of these things apart from the fact that there is evidence going on in the cell. But as causation and as the unifying explanation, I'm very skeptical about the prion hypothesis. And if I'm wrong, great, I'm wrong, then we can move forward. But no one's convinced me I'm wrong so far.
Well, yeah, my second guess to say no, so that's reassuring.
I could have given you a shorter answer, sorry.
Yes, yes. Well, I asked Parkinson's disease, and kind of just no, it's not. Okay. Coming to the clinical part of the diagnosis, we do the EMG and the others. The other thing is blood test. And I just wondered, there's so much about neurofilament light protein out there. Is that likely to help us in the future?
Well, it's helpful in specific ways. So your neurofilament level, broadly speaking, it correlates with rate of progression. Now, I don't think knowing the neurofilament level is better than a neurologist with a lot of experience looking at a patient in the way I described earlier. So that's not necessarily an advance, although people who like tests and perhaps don't have the same level of experience might latch onto this. And in fee-paying systems, if there's a test to pay for, then it gets taken up. So, but I would be very cautious if you are a non-expert neurologist or even a non-neurologist thinking that you can take a blood sample, measure the neurofilament level and tell somebody what their survival is. I think that is going to be a very inaccurate process because there are so many different things, and I don't think it trumps clinical skill. We are using neurofilament as a marker of responsiveness to drugs. So the one drug that does, I think, clearly have an effect, which is the antisense against SOD1, it's very clear that the neurofilament levels in the patients given the drug dropped within 3 to 4 months. So it's an early marker of something which can affect the disease trajectory. So we are using that in experimental medicine studies where we're trying drugs in individual or in groups of patients and looking at their neurofilament levels to try and screen drugs that are really worth taking forward. So in that sense, I think it's extremely helpful. The next question is, well, could it help you diagnose the disease earlier? So if you went to your GP and you just said, I've got a stiff hand, and obviously 99% of people with a stiff hand have got arthritis or something. So We looked at all our clinic patients who've been referred to us at first visit, and we looked at their neurofilament levels, so the early patients, and patients in whom there was a diagnostic question. And we asked ourselves, blinded, do we think they have motor neuron disease? Definitely, possibly, or definitely not. And we scored them. And the neurofilament level did not contribute to our accuracy at all as experts. The next question, does it contribute to, GP's ability to fast track a patient to clinic? That study has to be done, and it might do. Can it pick up people preclinically? Is an important question. So if you have a genetic mutation and there's a treatment, we need to know when to start that treatment, because you can't start treating 10-year-olds with SOD1 mutations. And the evidence seems to be that the neurofilament level probably rises about a year before the patient would otherwise present to the clinic. And that is not soon enough. If you really want to intervene to prevent disability, you need something else. So neurofilaments obviously is a sign of neuronal damage, axonal turnover. So it's already a sign of irreversible damage, even if the patient doesn't realize there's something wrong. So it's got lots of value, neurofilament, but then we need better and earlier biomarkers.
And it's kind of a generic thing, isn't it? It's not disease specific. Is it likely in the future it will be specific for diseases?
Probably not particularly, but so it's not disease specific, but if you look at all the diseases where neurofilaments go up, HIV, encephalopathy, and ALS have the highest levels by far. So, it's distinctively high. So given the appropriate clinical presentation with higher neurofilaments, I mean, that is suggestive that this is ALS rather than some other disease. So it's not very useful in Parkinson's or even Alzheimer's. So it's to do with the rate of progression, probably the long axons that are involved, I don't know. So I think what we might get with time is combinations of biomarkers that are more disease specific. So we are looking now at lots of preclinical samples with deep proteomics to see whether there's anything that is a pattern that we recognize. We might see tests like, so, for prion disease, we use RT-QuIC. So this assay where you shake up the serum with some seeded aggregate protein, and then you get a protein aggregation. And so we're trying that with TDP43. So it's possible that we might see some changes in that early. And then very specifically, TDP43 functions as a factor in splicing. So if TDP43 is leaving the nucleus, you get splice products, so-called cryptic splice products that are not physiological. And so if those can be picked up in the serum, they would be a specific test of TDP43 proteinopathy. And that is something that's potentially of great interest, but it needs a lot more work.
Okay, some hope. And then the misdiagnosis rate of MND, 30% by non-specialist. That should non-specialists be making a diagnosis of MND?
No, definitely not. There are a number of reasons why you shouldn't make the diagnosis if you're a non-specialist. It's one, because you can get it wrong, and two, because you don't know what to do next. And then if you say to a patient, well, you've got motor neuron disease, but I don't know what the answers to any of your questions, that's a distressing, you can cause immense distress by that, because then the patient goes home and spends a couple of unhappy hours on the internet. So I think if you make the diagnosis, you need to have a plan to support people. That's fundamental. And, I've had terrible examples over the years of people who just, because they weren't thinking, and also they thought, oh, that's motor neuron. I haven't seen that since I was a student, or maybe I've never seen a case. And they were kind of, yeah, oh, that's what you've got, that sort of triumphalism in diagnosis. And that mistake, can cause a lot of misery.
With John Zajiczek and Oliver Hanneman, I did an epidemiological study of MND in the Southwest, Devon and Cornwall. And at the time, I was reading a lot of the databases, and there was almost a uniform 5% misdiagnosis in many of the national databases. I'm assuming those are non-specialist diagnoses. Is that?
I think it's very difficult to know, when you say misdiagnosis, then you have to ask who's making the diagnosis, what's the denominator population, and all the rest of it. So I think misdiagnosis in neurology clinics is relatively rare, actually. So I would put it below 5%. I think most neurologists are perfectly capable of diagnosing motor neuron disease in most cases.
Yeah. And the closest misdiagnosis you did mention was multifocal motor neuropathy.
Yeah. Well, I think that, that people are fond of producing long lists of so-called mimic disorders. I think this is pretty unhelpful, actually. In almost all of those cases, there were plenty of clues as to sort of a disease being present. So if you've got typical symptoms and signs of ALS and you're progressing, there are very few other things it could be. The most difficult is there are 3 or 4 things, I think, where consistently people get this wrong, and conduction block neuropathy is one. Inclusion body myositis is another, and also structural disease, of the spine. So cervical spondylitic minor radiculopathy. Of course, every patient in their 60s will have an abnormal, or probably a normal MRI for their age, but it will contain various things that people want to blame for their symptoms. So that can occasionally be wrong. But otherwise, there are very few, in the appropriate setting for most people with MND, there's usually no diagnostic doubt on clinical grounds, actually. So an EMG, in fact, is a kind of hand-holding exercise a lot of the time.
You did talk about this dilemma in the very early stages where the diagnosis is not quite certain. You suspect it, but kind of is still too early. And you talk about the dilemma. Should the patient be told the diagnosis that is not yet possible, or that MND is one of a number of possibilities? What's your practice? Do you pop the name out early?
I think I wrote that, a long time ago now. And my practice now is just to be very straightforward with people. I don't think there's anything to be gained from, the patient deserves to know what I know. As long as it's quite clear that that individual thinks like that. I mean, of course, this is about being sensitive to people's individual need for information. So there are occasions where it's quite clear. So somebody comes on their own and they have no idea there's something serious going on. And, I know that sitting them down and telling them this is motor neuron disease, or it could be motor neuron disease, in an unsupported environment isn't really very helpful. So in that sense, I sometimes do say, well, we're going to do some tests and we're going to see what happens over a period of the next few months and see if things change. And sometimes people go off perfectly happy. But the majority of people, I think, have already got, in their head that something serious is happening. So for me to try and, be neutral about it doesn't make any sense. So I have to acknowledge that this is a serious problem, that it may involve irreversible loss of function, and we go through the possibilities. The difficulty is, when you've got somebody with a single limb involved, for which there's no electrophysiological support anywhere else, and particularly if it's lower motor neuron predominant in that limb, well, I am a bit cautious there because I have seen patients who just don't progress and they end up with some weak limb for reasons I don't think I understand. But that's the scenario that's most difficult. I mean, it's relatively rare, but in a specialist clinic, you see this, on a regular basis. I think when someone's got a mixture of upper and lower motor neuron signs in a limb, unequivocal, so you have to have quite significantly brisk reflexes compared to the other limb plus wasting, then I think that is likely to be motor neuron disease in the absolutely overwhelming majority of cases.
And concerning breaking the news, from what you said, clearly should be with the family around. What other factors do you consider when you're breaking the news of MND to the patient?
Well, I think, it's got to be about that individual. You need to understand that person's worldview, their emotional state, their support networks, what they want to know. I mean, and people are very, very diverse, I mean, increasingly people just want straightforward conversation, but there are still people who say, I'm going to deal with this by waking up every morning and just dealing it all day. And I don't want to have any understanding what might happen next month or in 6 months' time. That's what they say. And I have to accept that's what they believe. I think mostly you have to explore that a little bit more carefully, because if people are saying, I'm frightened, so I want to understand what's what I call the semiology of these kind of statements. What is the patient saying to you? They say, I don't want any information. Since that's so unlike most people and most human beings in most situations, I mean, generally speaking, if we have a problem, we want to find out the cause of it, right? Whether it's our car or our arm. So if someone says, I don't want to know anything, then I think what they're telling me is that they are terrifically scared. And I say, well, I understand that why people are scared, and that maybe it's better not to know things. But actually, usually when I've told people what I know, they feel less worried than they did before. So if you're telling me you don't want to know something, that means that in your head are some terrible things that you're very worried about. So why don't you tell me what's the worst thing that you're worried about? What's the worst thing that could happen? And then they say, well, I could suddenly die suddenly and choke to death, because that's what people think happens. And I can say, well, in 2,500 patients, I've never had a patient who's choked to death suddenly. It doesn't happen. And so, that's how I kind of deal with it. I try to understand what that person is really most worried about and deal with that, and leave the patient going away with a slightly better view of a terrible situation than they had before.
Okay. Thank you very much for that. The issue of the terminal stages comes up, of course, and the use of ventilation to keep people with ALS alive. I know that in the UK and many parts of the world, that's kind of not encouraged. But I think in Japan, it's kind of something. I think it's something standard there. Is it a cultural thing or is it a medical thing? Trying to avoid with
No, I
MND.
think it's very cultural. If you look at what's happened in Japan in the last 20 years, they have their rate of invasive ventilation, tracheostomy ventilation, has gone down very considerably.
Okay.
And the reason for that is that the success of non-invasive ventilation, has really demonstrated you don't need a tracheostomy, and a tracheostomy increases the burden care very considerably. So our approach to this is that, we're not there to prevent people having invasive ventilation. We are there to try and understand what they're trying to achieve as a human being. So you need to understand what's in that patient's head. What's their expectation? So we've had some very interesting responses to that question.
Mm.
People say, well, look, I'm getting weaker and weaker, but I want to see my children grow up and go to university. So just, make a hole in there, put me in the corner, and I can watch the world. I'll still be there for them. And then when you explain, well, firstly, the disease will continue to progress, and that having a tracheostomy does not necessarily guarantee survival. So in fact, in settings where large numbers of tracheostomies are done, so Italy is another country, the survival rate is on average 1 year from tracheostomy insertion. And that's because of complications and infections, but it's actually fundamentally because the disease is still progressing. So, there are all sorts of, eventually in an aggressive neurodegenerative disease, you will get loss of cardiological reflexes, the fundamental respiratory drive, and all sorts, and cognition. Not in everybody. So I think the point being, simply plugging somebody's breathing with a tracheostomy doesn't guarantee long survival. It does not actually turn out to be the best way to palliate their symptoms. Non-invasive ventilation is a very efficient way of doing it, and that's freely available now. So the need for tracheostomy has gone right down anyway. It's not solving any problems. Now that will change, though, because if we have treatments that prolong the disease, then I think the whole thing will gradually change. So there are some patients for whom tracheostomy is entirely appropriate. And those are typically people who have got slowly progressive disease, but are in a state of significant disability, who have a very strong and well-constructed support network, who are highly motivated, and who don't get cognitive impairment. Now, patients like that can do quite well with tracheostomy, as long as they really need it. If they have it put in when it's not necessary, then it just makes their life immensely more uncomfortable and complicated. So, these are the conversations that we have with people. And we've only had 3 or 4 people have a tracheostomy, because actually most people realize that what they want, which is to live on in some form, isn't going to be what they think it is. So they realize that. And secondly, you've removed the fear of this choking as well as another thing. So, I think an awful lot of people, when you have a straightforward conversation, which is supportive, open, and it's not meant to be, you can't have a tracheostomy because the NHS doesn't do that. That's the worst possible thing you can say to somebody, because it's not true, but it's also, creates this sense that someone is denying me something that will keep me alive, which is a terrible concept. And that's not what's going on. So it's about having that conversation and using your experience to explain what really it's all about.
And still at that kind of in that zone, the issue of euthanasia, I think there was one or two high-profile cases of people going off to Dignitas. What's your experience of that? Have you had any patients wanting to?
Yeah, we've had about 20, 25 people go to Dignitas, maybe more. But so, what we try and encourage is an open conversation. So the worst thing is, and this has happened or happened a lot, it happened early on, people felt, well, they couldn't discuss it with us because we'd get into trouble. That's what they often said, which isn't true. And you'd suddenly hear that somebody had gone off and their relative would turn up with the wheelchair and say, we don't need this anymore. And that was very shocking because we felt, well, we weren't there for them when they made that decision. So we didn't bring it up as such, but we use every opportunity to understand whether people are worried about the end of life, what their decisions are. And if they want to mention Dignitas, we want them to feel they can do that without any fear. Now, we don't have any communication with Dignitas. We send our letters to patients, so they have all the information they need. We respect their decisions. We don't have a view as a clinic because that would be wrong. Within the clinic, there are probably a whole range of views about this, like there is everywhere else in society. We want to be open and supportive to everybody. What we most of all want is people feel able to actually tell us what's on their mind, because then we can help them.
And perhaps on a positive side, statistics you cited showing 10% of people with ALS will live beyond 10 years. I'm sure must have
maybe slightly less than that, but it's in that sort of region, maybe 5 to 10%, yeah.
And is that across the board with all forms of MND, or is it just this primary lateral sclerosis, which we know?
Right. Well, it's different types of disease. So it's not just primary lateral sclerosis. And people with primary lateral sclerosis essentially can live close to a normal lifespan. But, if you're getting it in your 50s or 60s, then, your life expectancy is 80 to 85. And many people with PLS will live for 2 or 3 decades after the diagnosis. Not all of them. But what you see in people, that population of people who are alive 20 years after the onset, they're very varied, actually. So there isn't one pattern that predicts that longevity. It's more to do with that multidimensional kind of effect, the rate of progression, the absence of respiratory involvement, the absence of cognitive involvement, the lower limb, lower motor neuron predominant. All of these factors are more present in that long survival group. They do vary.
You did give an anecdote of Mary, who was given a 2 to 3 year prognosis and ended up living 25 years, perhaps beyond the lifetime of the doctors who gave her the prognosis. Is the moral of that story not to give individual prognosis, or to be less specific about time?
No, I don't think you can use these extreme cases to govern most of your practice. That would be a mistake. If there's any moral, it's to be straightforward with people and say, well, I'm using my experience to answer your questions that are important to you. And if people want some kind of prediction of their life expectancy, we will give them one. And in 95% of cases, we're correct, actually, because of our experience. But we always say, well, there are some factors which you can't really account for. I think that it's most likely that you're going to, your disease is going to carry on between 18 and 24 months from now. And that's a 6-month window. So it's, obviously some, and that what I'm telling them is that you haven't got a disease that goes on for decades. That's what they're really asking.
Yeah.
So I say, well, I could be wrong, because occasionally we just hear that people have died suddenly in their sleep peacefully, and we don't expect that happens. Occasionally people will live for longer for complex reasons, but a lot of it's governed by respiratory involvement. So that's the kind of conversation we have. We tell them what we know, what's in our head, as long as that's what they were asking us.
Okay. And how much does the shortened life span of people with ALS restrict how much you can do in research? In other diseases, they kind of have a cohort they can follow for 10 years, 15 years, but
no, I don't think it. I mean, there are 2 ways of looking at it. One is that because it's happening, the natural history is compressed into a short period, then you've got the best chance of actually understanding it, because studying people who've got very slowly progressive disease that goes on for decades, well, that's more difficult. The alternative is to say, well, it's so terrible in terms of its pathological aggressiveness that then is a very difficult challenge to reverse that. But I think it's actually not a problem that it's a short disease in most people at all. And I think increasingly our efforts have to be about prevention, early disease, and I think what happens once people have got very significant disability is an irreversible problem on current evidence. So we should be focusing our efforts on trying to find drugs that work early and even before the condition starts.
And what would you say are the major uncertainties in the field of ALS at the moment?
Well, I think, in terms of causation, we are increasingly understanding the genetic architecture. So that is becoming gradually a more, informed area, but there's still lots to understand. Why somebody gets ALS, who gets ALS rather, if it's defined genetically, we need to complete that genetic architecture story to understand why people with so-called sporadic disease, what is the genetic contribution? So that's an important task. But actually, we know far less about the biological basis of all this variation that we've been discussing. Why some person progresses rapidly, some person slowly. We have no real handle on that. If you understood that, then I think you might well have some quite tractable treatments. So we are putting a lot of effort into trying to understand whether we can compartmentalize the disease and say, well, there are 4 types of ALS based upon a biological signature. One's more inflammatory, one is more metabolic. I mean, these are all just speculations, but we might find that there's nothing that allows us to separate fast and slow progressors by any kind of biochemistry. But I suspect that there is a biological underpinning for this. So I think that's a very important task that we are currently very focused on. I think why the initiation is the key thing we don't understand. Why does it apparently start in an anatomically focused way? What are the events that lead up to that? If we understood those, you could argue that you could prevent things from propagating in any kind of a way, whether that be prion-like protein or some loss of some oscillatory network equilibrium in complex motor circuits. I mean, there are a number of ways of thinking about it. And then we need to understand at the cellular level where the real problem is. Okay, so obviously, if you have a motor neuron, if it loses its synaptic contact, then that shows over, whatever it looks like under the microscope. So the synapse is clearly very important. So I think targeting synaptic integrity is ultimately very important. But upstream of that are things like axonal transport, mitochondrial function, oxidative stress, etc., which might be driving the loss of synaptics. So understanding the order of events, I think, in the cell, if that is even possible, is an absolutely key challenge, because there's lots of phenomenology, lots of there are 10, 20 different pathways that are disturbed, but which one came first? If we understood that, then I think we would be in a different place. There may or may not be an answer to that question, because if there are 10 points of entry, it could be that it can enter in any of those 10. So there's no first pathway. But I want to understand the early changes and not put drugs focusing on late downstream changes. That's just not going to be helpful.
Kind of similar to what happens in cancer, where they're trying to find out what's going on with the first cell, I presume. How much can we translate from the genetic forms of ALS to the sporadic, if you learn so much about SOD1, MND?
Well, yeah, that's a really important question. And I think what's very clear now is that SOD1 is not a particularly translatable form of ALS, which is a big lesson. Only 2% of people have a SOD1 mutation. Their pathology is different. They are now, at least potentially, can be treated with antisense. The other 97% or so of patients have TDP-43 proteinopathy, whatever the cause of their disease. They look fairly similar pathologically, or they're not always identical. So that argues that whatever's going on at the cellular level must be similar in those people. So somebody with a TDP mutation, is not distinguishable from somebody without a TDP mutation. Their brain and spinal cord look very much the same. So that at least offers the hope that if you use those patients as a model by creating models in a laboratory of those mutations, you're going to learn something important about the disease. But it's not by no means certain that that's the case. That is simply a tractable way into the problem. It's no guarantee. And it's just possible that, most people with ALS are not being properly modeled at the moment at all, and that's a worry.
And at the time you wrote the book, the C9 of 72 gene had not been discovered. Has that advanced our understanding of ALS?
It's massively advanced our understanding because about 10% of people with ALS have a mutation in C9 or 72. So it provides a group of people who can be studied who've got at least one biological cause that is homogeneous, although it also includes all of these different cofactors like aging. So we are building a national cohort of people carrying this mutation who've been identified because a first-degree relative had ALS or FTD with a C9 mutation. And we are going to follow them for years. And we're going to have 400 or 500 people in this group, and we're going to look at them and see what is going on before the disease starts. So that's our best hope of finding some marker that is detectable years before disease onset.
Yeah, that's one positive thing, at least. Are you planning to do a second edition of the book?
I think the answer to that is no.
Okay.
Partly because I don't have the capacity at this stage to do it. I mean, not because I'm falling apart necessarily, or maybe that's happening too, but I'm just so busy. I think it still is relevant mostly. It could be revised. I don't think the publishers are inclined to revise it. So that's another factor. We did another book called the Oxford Motor Neuron Disease Handbook, which is more for healthcare professionals, and we are revising that. But I'm coming towards the end of my clinical life. And so I don't think I should even be doing this. Actually, somebody else might want to do it and give their perspective.
Should we be looking forward to a memoir then?
A memoir? No, I don't think. I think that would be tremendously boring. Well, I mean, if I manage to find the time and the energy, I think there is a book to be written about neurodegeneration for the general public, the intelligent lay reader, if you like. Yeah. Why do we have diseases of the aging brain at all? I mean, I don't think we think of age-related, well, maybe I'm wrong about this, age-related gut dysfunction or age-related, I mean, the things that happen in aging to all of the organs, right? But this process where an organ in a very stereotyped way falls apart, that's a rather nervous system specific problem. And why is that? And it's to do with human evolution, human complexity. How do we approach this problem scientifically, clinically? What are the stories behind all of this? I think there's an interesting book along those lines, but whether I'll ever write it, I do not know.
I will be looking forward to reading it. Do you have any books then you can recommend, either on the brain or this theme of ALS? I usually ask for 3 books.
I think that books are a problem these days, aren't they? Who reads books anymore?
I do.
I mean, lots of people do, and you've got a nice lot of books behind you. So clearly people do, but I think textbooks are a problem because the flow of information is so rapid. If you want to know something, you're much better off looking at literature, a review article or something. So, but there are a few books which I think stand the test of time because they are basically classics and that are useful. So I don't think I've prepared my answer to this question very well, but one book that I would still pick off the shelf was written by Jan van Heijn, who was a neurologist in Utrecht in the Netherlands, and it's called Mononeuropathies. And it is the best book, for dissecting out somebody with, the difference between if they come along with a problem, is it a mononeuropathy, a radiculopathy, or something else? And it's beautifully written and lots of great examples. That's a fabulous book. I would recommend any training neurologist, for example, should carry that book around. Age the Examination of the Peripheral Nervous System is another classic book, which is, so getting good at examination through those books is very important. I think, it depends what you mean. There are books that have been written by people with ALS, who have told their story, and some of those are probably worth considering. There's a book called Rowing Without Oars, which is probably the best account I've read. I haven't read many books like this, but it's a beautifully written book about a woman who, a young woman in Sweden who gets ALS and how things happen. And that's quite moving and informative and human. So that's probably what I would say.
Thank you very much for those recommendations. And Kevin Talbot, thank you very much for participating in this episode of the Neurology Land podcast. I'm most grateful. I've enjoyed and learned quite a lot. To my audience, thank you for listening or watching the podcast. If you found it enlightening, you might want to check the book. You might also read my small book called Surfing the Brain, or check my website called Neuro Checklist. And to make sure you don't miss the next episode, subscribe to the iTunes, Spotify, or YouTube channels. Until then, I will say bye-bye. And Professor Talbot, thank you very much for your time. I'm most grateful.
It's been a pleasure.